and I hope things are going very well for all of you. And again, I'm working here in GB Band Institute, and because we are under Ministry of Environment and Forest, so our mandate is basically work on environment and forest related issues. But before that, before I did my PhD, I worked on nonlinear dynamical systems as well. So I was working on small order, low order nonlinear dynamical systems that can be used for monsoon predictions. But today the presentation is something different, and I think I can share the presentation from here.
Yes, sir. Yes, sir. Yes, sir. You can share. I think in the present now. Yeah.
Yes, I think I can share it from. Oops. Share it now. But it doesn't show actually. I think just a minute. It is. I hope you can see the presentation right now. Is it? Can you see the presentation from there?
Yes, sir. Yes, sir. It's visible.
It's visible. It says ordination methods in ecology, applications on PCA and NMDS in R. So this presentation, I prepared this presentation few weeks back for some of the graduate students in our country, in our place, where they were first-year PhD students, mostly first-year PhD student, and they were working on ecology. So, for those of you who doesn't know what is ecology, ecology is basically the subject which deals with biotic and abiotic factors. And most of the ecologists in India, and as well as people from abroad, what they do, they collect plant information. And using that plant information, they correlate that information with different environmental parameters. And by doing so, what you really want to achieve is, you want to know what plant is available at what environmental condition, and how we can propagate these different types of plants. Plants, I mean to say, it includes trees, shrubs, herbs, and even bryophytes and pteridophytes. But again, I just want to clarify that I'm not a biologist, but the methods that I'm going to discuss in this presentation, they are all equally applicable to my science, that is in hydrology as well as in atmospheric science. So the rationale for this presentation is, these days we are working more on multidisciplinary approaches, where we talk mostly about interactions between systems. Nowadays, it is comparatively hard to work on a single system. I mean to say, on a pure hydrological budget or on a pure atmospheric process, because they are all coupled with each other. A hydrological system is coupled with an atmospheric system, and then the atmospheric system is coupled with the biological system. So the interactions between these systems, they play a very crucial role for assessment of basic questions. For example, I mean, most of us, I just give you one example, that we really do not know how much water is extracted out by different varieties of plants across the world. There are some information available for certain plants, but not for all the plants, not for all the trees. Now, when we do not know the amount of water is being extracted out by different plants and different shrubs and herbs, we really, it is comparatively hard for us to come up with a pure water budget. And once we do not have a pure water budget, it is always harder for us to predict atmospheric processes, because water budget plays a crucial role in terms of evapotranspiration and rainfall. Therefore, the interactions, identification and assessment of interactions are nowadays a priority area. Because I'm working in Himalaya and in Uttarakhand, where most of the state is covered with biological it has a very high biological diversity. Around 50 to 55% of the state is covered with forest. For us who are working in Uttarakhand, we are typically working on issues that is related with forest and interactions of forest with biological, with hydrological as well as atmospheric processes. So therefore, to understand ecological systems, there are many mathematical and statistical methods and tools available. I'll just highlight two methods of ordination, what we can use for assessment of these different components. So before I go into the detail, the presentation is a compilation of many resources that we have collected, mostly from Oklahoma State University, Department of Biological Science, University of Montreal, and then there are many online resources. And the very important source that I have used is from Department of Statistics, University of Pittsburgh. Now, those of you who are interested in ecology, I would suggest you to go to this webpage. This webpage gives you much detailed information on how to use different methods and what is the rationale of these methods and what you can extract out from these methods. Now, I'll start with community ecology. Now, what is community ecology? What community ecology is? Identification of plant species or tree species in a group. See, when we say that this particular forest is a, let's say, cheer pine dominated or let's say sal dominated, we mean to say that that particular forest has a huge number of sal trees. Now, there are many methods from in biological sciences where you can go and simply count the numbers. Say within a 2-hectare area, if you say that that forest is sal dominated, what you do, you simply go and count every trees. It is a very kind of rigorous task. There are methods where you put up where you put quadrates of 100 meters or as well as 100 square meters or 10 square meters, and within those quadrates you calculate, you count number of trees, and based on that you come up with the community concept. Now, in community ecology is one of the challenging branches of the modern environmental. It is challenging because community ecology doesn't only mean of counting of trees. What you simultaneously do, you note down all the other environmental parameters as well. When you go to a forest, you count the trees, you collect soil samples as well as meteorological parameters like temperature, precipitation, rainfall, etc. And then what you do, you collect some geomorphological features as well. That means terrain, elevation, height. So all these informations that when you do community ecology study, you collect all these different information. So you come up with a very huge database. But unfortunately, most of these databases, they are in a situation where you cannot simply do a simple statistical method and you come up with a conclusion. These days, you need to link all these different parameters like environmental parameters, geomorphological parameters, as well as biological parameters and meteorological parameters. You have to bring all those information into one platform to come up with certain inferences. The biggest problem with the community ecology is some of your parameters will be in time series. Time series means you have the parameter varies with time. So your observations are time dependent. Let's say it can be daily, it can be monthly, it can be seasonally, it can be annually, or for us, for meteorologists, it can be even in hertz. That means for a single second you can have 10 number of values. So when your data varies so significantly in space and time scale, it is sometimes very difficult to bring them into one platform and analyze them just to infer whether a certain meteorological parameter is impacting some species count or not. But community ecology typically need to analyze the effect of multiple environmental factors, and that is why you collect all the environmental parameters. And therefore, on species, you collect all of your data on species, and then when you collect all your species informations, definitely the measurement has some structural error as well as measurement error. So when you do the community ecology data analysis, you have to also keep in mind that your data has some inherent error, and you have to come up with some ideas how you analyze your data, just neglecting or just doing some data analysis where your error footprints are minimized. Now, it is not surprising, therefore, that the ecologists have employed a variety of multivariate approaches. Now, again, from those people who are from mathematics background, you know, multivariate is definitely when you have multiple variables. So when you have a data set where multiple variables are there, and then in that case, you do multivariate data analysis. And because in community ecology, we have multiple data, multiple variables, we have to do multivariate data analysis. These approaches have been both endogenous and borrowed from other disciplines. I have told you before that these days, most of the sciences where we work on environment, it is in most cases, it is in a larger framework, it is combinations of different sectors. I mean to say, you cannot simply say that the plant only grow because of one parameter. There may be multiple different parameters from different perspectives, which is responsible for a plant growth. Therefore, for analyzing community ecological data, you can use some techniques which are typically earlier, which typically people were using for, let's say, meteorological problems. But nowadays, when you do community ecological data analysis, you can borrow those statistical methods and apply to your data. The majority of techniques fall into two main groups when you do community ecology. The first section is classification, and the second part is ordination. So I'll not go into the detail of classification. Classification of community ecology data is purely, in most cases, it is based on a biological classification. That means you have you analyze, first of all, you analyze your plant data in terms of, let's say, some species, some genus, some family, some order, and then based on that classification, you compile your environmental parameters. But it is a bit complicated. I think it will be better if biologists come forward and explain the classification methods. Rather, I'll focus on ordination method. Now, what is ordination method? Ordination method is an arrangement of order of species and or sample units along the gradients. Now, the gradients terms that what we are using here, it is not purely the mathematical gradients. Rather, it is the ecologists, they say gradient is like of different multiple locations. For example, let us assume that you are doing community ecological data collection from one forest. The forest has a location X, and at that location you have the forest community data. Now, if you go from location X to location Y, your elevation changes. In Himalaya, typically your elevation changes, then your slope changes, your aspect changes, as well as geomorphological structures, let's say drainage density changes, geology changes. So these are different things that changes with your change in location. So when you do ordination, your gradient term in ecology is actually representative of your change in location. Now, briefly, the history of ordination methods. Ordination methods actually, I mean, all the ordination methods that we are using these days in ecological assessment, they were not actually developed in terms of ecological data assessment. Initially, they were purely statistical analysis. It started with the work by Ramensky in 1930. He began to use informal ordination techniques. For example, mostly in vegetation. In 1951, then Curtis and McIntosh developed the continuum index. These days, people don't use continuum index for community data analysis. In 1954, Goodall introduced the term ordination in an ecological context, where they have used principal component analysis. So the history of principal component analysis is very old. I mean, we use PCA for most of our data analysis, but earlier in 1954, it was used for the first time. Then Bray and Curtis developed polar ordinations, which became the first widely used ordination techniques in ecology. And in Austin in 1968 used canonical correlation. Still now, canonical correlation is a very handy method for community ecological data analysis. And these canonical correlations, as well as Bray-Curtis polar ordination method, they are equally applicable to hydrological sciences. But I mean, it depends again with your problem, what you are going to achieve with your statistical methods. So it can equally be used from ecology as well as in hydrology and meteorological issues. In 1973, Hill introduced the correspondence analysis to ecology, and correspondence analysis gradually supplemented the polar ordination. So polar ordinations people don't use these days. And Kruskal's 1964 non-metric multidimensional scaling is rapidly, I mean, people started using it from early 1970s, and still we are using this non-metric multidimensional scaling. Here in G.V. Pant Institute, as well as many biological institutes across India and across world, people still use non-metric multidimensional scaling. And recently we have also started using this method for water-related issues. Now, just briefly, these correspondence analysis were again updated by Hill in 1979, and then this detrended correspondence analysis was developed, and it's still being used. And for those who are interested in community ecology, I would suggest you to read that book called Multivariate Analysis in Community Ecology. It is kind of the mother book when you do ecological analysis. You should read. I mean, we all should read this book when we think of doing community data analysis. And finally, the canonical correspondence analysis is also another method which was developed by Tabrak in 1986. But this method is again very rapidly used in today's world. Now, before I go into the detail into the statistical methods, there are some properties of the community data. Community means, I mean to say, in terms of biological community, plant community, trees community, shrubs and herbs community. Now, all of your data analysis and data interpretation depends on how you come up with your database. I mean, here you can see that on your right-hand side, there is a table. These are basically scientific names. My apologies, these names should have been in italics. I mean, I couldn't do that. And these are different locations. These are different plant species, sorry. And these are bryophytes, and these bryophytes, you can find them over some plant species, and these are their abundance data set. Abundance is one kind of it is a kind of parameters by which you can say how much it is available or not. It is linked with importance values. I mean, here it is importance values, but it shows one kind of abundance. Now, in ordination method, essentially operations are on community data matrix. So when we do ordination, we do that on this kind of data set. A community data matrix has structure, usually species that I have shown you. These are the species as rows and samples as columns or vice versa. So these are the samples. Now, when you do your data management in our institute, as well as in Wildlife Institute of India and BSIs and JRSIs, what we do, we use these species as our columns and locations as our rows. The term sample has diverged from its usages in statistics and refers to the basic unit of observations. I have told you before, in ecology, sample is more of like quadrates. If you are analyzing data on the basis of quadrates are nothing but some plots. You put a plot of 10 meter by 10 meter or 100 meter by 100 meter, and within that plot you do the community analysis. So that is how you define samples in community ecology. In most studies of vegetation, the sample is a quadrate or transects. Yeah, it can for the complex terrain, it can also be as transects. Let's say you go along a elevational transect. That means you start collecting sample at an elevation of, let's say, 100 meter, and then for each 100 meter sampling interval, you go and you collect all your data, and then you reach up to 4,000 meter elevation, and you do this analysis for multiple transects across the Himalaya. You start from Jammu and Kashmir, and you take different transects, and then you reach to Uttarakhand. So each of your transects can also be considered as samples. Now, there are some properties of community data. So when you collect these ecological data, the most important parameter that you're looking for is abundance. Abundance is simply, it means how much abundant is that is a particular species. In general term, this abundance can refer to density. It can also be linked with biomass, cover, or even incidences. That means some species are very typical to one place. Near Nainital, there is one species called Padua. Padua is a small tree. It is very good for land rehabilitation, because most of the Himalaya, we are facing this land degradation problem, and Padua is a potential plant which can be used for mass replications, which can be used for soil degradation. But you find Padua across the Indian Himalayan region to a single location only near Nainital, a place called Padua Down. So So when you do a survey for Padua, you'll only find it only on a single locations, and that location can be considered as an incidence. Now, the choice of abundance measure will depend on the taxa and the question under consideration. That is also true. I mean, what you want to achieve with all of your sampling, all of your data collection is also an important factor. Any of the matrix constitution column vector is considered as the species compositions. I have told you before that your column can be species composition or it can be a location. For us, we use it as a species composition. And then it's frequently expressed in terms of relative abundance. I mean, if you have very high species composition, you can go up to 100%, and if it is very low, it can be 1%. The purpose of ordination and classification method is to interpret patterns in scientific composition. Now, what are the general properties of community data? The community data, they tend to be sparse. Sparse in the sense, see, I mean, when you have some tree data or some species data with you, you can see that that particular tree or that particular species is sparse across a wide region. It is not, in most cases, it is not concentrated to a single location. If a species is concentrated only to a single location, irrespective of it is plant, herbs or shrubs, or even for a biological species, it could be categorized as endangered because you will not find them easily to other locations. But it is not the general trend. In most cases, your data will be sparse. Most species are infrequent, implying the majority of species is typically present in a minority of location. Although it can be sparse, but it may be concentrated at a single location. See, in Himalaya, if you come to Uttarakhand as well as in Jammu Kashmir or let's say Himachal Pradesh, in most cases you will see cheer pine trees. Pine trees are very common in northwestern Himalaya. But oak trees, which are broadleaf trees, they have much larger socioeconomical as well as hydrometeorological value. They are found on a certain location. They are mostly found on northerly slope because northerly slopes are colder, and they are found and they are associated with water bodies. I mean, if there is a small dhara or a small nola, you find them over there. So the number of factors influencing species composition is potentially very large. That I have already told you before. It is true for any physical processes. Let's say if I say rainfall is itself is a very complicated process. I mean, it doesn't depend on a single parameter. It depends on multiple parameters, and that is true for biological system as well. Composition of species or a particular species species distribution does not depend only on a single or two parameters or three parameters. It may depend on multiple parameters. Although it may depend on multiple parameters, there will be very few parameters which are actually having larger impact. So when we do ordination, our aim is to find out that 3, 4, or 5 parameters which are actually having larger impact on species distribution. That is true for many of our hydrometeorological processes as well. When we say, let's say, rainfall is dependent on many things, what we try to do, we try to find out those parameters which are having larger impact. For example, India Meteorological Department, they use monsoon forecasting, and for monsoon forecasting they use artificial neural network. But that artificial neural network model the India Meteorological Department is using, they have around 8 to 16 parameters. But if you go through the monsoon rainfall processes, you can find out, let's say, more than 20 or 30 odd parameters which can directly or indirectly affect rainfall. But for monsoon, only there are 8 to 16 parameters which can have real impact. So the number of important factor is typically low. That is, a few factors can explain the majority of the explainable variance. Another way of saying that it is the intrinsic dimensionality is low. The other factor, the other property of the community data is noise in the data can be substantial. Because, see, I mean, noise can be of many types. Errors can be of many types. It can be random error. It can be systematic error. For community data, because we do collect data manually, in most cases, the error can be large because we did not identify the plant species properly. And then that can be also an error. And then it is because it's most cases it is count like 1, 2, 3, 4, 5. Like in one coordinate you find 10 number of species A, or let's say 5 number of species B. If your count is I mean, when you note down those counts, if it is somehow you have done a mistake, it will propagate, and the noise in the data will magnify. This is large due to stochastic events, though observer error may also be appreciable. There is much redundant information. Species often share similar distributions. So that is also a very important factor in community data. So if you say that, let's say species A is found over a particular transect or a particular elevation, because species B depends on species A, you can very easily say that if species A is available to a certain location, species B will also be there. So this is a very common property of community data as well. Now, when we have such different type of data set in community ecology, we use ordination. Now, why ordination? Ordination is primarily endeavors to represent samples and species relationship as faithfully as possible in low dimension space. So that is what I was trying to say from the beginning, that when we do ordination methods, we reduce the dimensionality of the data. In physical processes, we know there is a term called degrees of freedom. So these degrees of freedom, very crudely, these degrees of freedom, they represents the number of parameters that you need to define a process, and in natural system, these number of parameters can be large numbers. So we cannot simply visualize data in n number space. So we need to reduce the dimensionality, and we reduce the dimensionality to low dimensional space, and that is what we do using ordination method. It is impossible to visualize multiple dimensions simultaneously, and therefore you do ordination. Again, ideally and typically, dimensions of low dimensional space will represent important and interpretable environmental gradients. A single multivariate analysis saves time. I mean, there are many methods. You'll show in my subsequent slides, there are many methods by which you can do multivariate analysis, but a single method where you can reduce the dimensionality of the data and you can very easily picturize the data, it saves time. And then it can also be easy for us to for interpretation. The graphical results from most techniques often lead to ready and intuitive interpretations. When you have very easily interpretable graphics with you, you use that data very easily. And here I have tried to give you some example. The main example of ordination. The main objective of ordination is to display the objects which are often sampling sites in ecology into multivariate dispersing diagram. Now here you can see these two diagrams. One is from sweet to sour, and the other is from difficult to easy. Sweet to sour is the sweetness to sourness, and difficult to easy in terms of feeling. Now I have put banana far away, but it is very close to easy, and it is very close to sweet. Now the 0, 0 values indicate that it is not, I mean, it's very close to 0. It's neither sweet nor sour. But as you go on towards sweet direction, you find it is sweet, and if you go towards sour direction, you find it sour. So it's a 4-dimensional plot where peeling and sweetness is represented. For example, in case of pineapple, you say that it is very hard to peel, but it's kind of sweet. It's very close to sour, but it's kind of sweet. It's not exactly sour. So that is the essence of ordination method. So when we do ordination method, we come up with these four dimensional plots, and using these 4-dimensional plots, we analyze, we interpret our data. Now, what are the different types of ordination? Now, the types of ordinations are indirect gradient analysis and direct gradient analysis. In indirect gradient analysis uses only the species by sample matrix. Information about the environment is used after indirect gradient analysis for interpretations, and we are essentially asking the species to tell what are the environmental parameters that you actually prefer. But for direct gradient analysis uses external environmental data in addition to the species data. So when we do direct gradient analysis, it is more of like regressional analysis. Direct analysis allows us to test the null hypothesis that species composition is unrelated to measured variables. In terms of indirect gradient analysis, again, there are two different methods for indirect gradient analysis. One is distance-based approach, and the other is eigen analysis-based approach. Now, I have I have categorized into two different approaches. One is distance-based approach and the other is eigen-based approach. But mathematically, they are kind of fuzzy. I mean, actually, eigen-based approach can also be used in some cases for as a distance-based approach, and vice versa. But still, they are different. And I cannot. I mean, because of time, I cannot go into the detail of why and how they are different. But for distance-based approach, the methods are polar ordination, principal coordinate analysis, and non-metric multidimensional scaling. For eigen-based approach, we have principal component analysis, correspondence analysis, and detrended correspondence analysis, of which currently ecologists across the world, they use non-metric multidimensional scaling, and principal component analysis is again being used. But I mean, currently most of the ecologists across the world, they use correspondence analysis and detrended correspondence analysis. But unfortunately, in this presentation, we will only talk about non-metric multidimensional scaling and principal component analysis. Because I know that people are from different backgrounds, I have tried to minimize the mathematical interpretations and notations. I'll only use very simplified example to show you how PCA works and how non-metric multidimensional scaling works. Then on, I'll show you some R codes, how you can use R. It's an open source software to analyze your data set. Yeah, the third thing is the second part is direct gradient analysis. I will not go into the detail. It will probably take again another two to 3 weeks just to go into all the methods. But there are methods like linear model, redundancy analysis, unimodal models, canonical correspondence analysis, and detrended canonical correspondence analysis. These are parts of direct gradient. We'll only focus on indirect. So commonly used ordination methods, yes, again, PCA and CA, that is principal component analysis and correspondence analysis, where eigen-based approach is adopted, and the other is principal coordinate analysis and non-metric multidimensional scaling, where distance-based approach is adopted. In these cases, we calculate dissimilarity and we use different types of variables. So I'll show them in detail in my subsequent slides. So I'll start with principal component analysis. So principal component analysis is an ordination method preserving the Euclidean distance among the objects, and PCA is only applicable to multivariate quantitative data. Again, I'm representing PCA in terms of ordination method of ecology, but in my paper, in 2 or 3 papers, like the one where we have studied Nino phases for Indian monsoon, where we have again used PCA. Simultaneously, I have used PCA for rainfall intra-seasonal oscillations identifications as well. So PCA is a very handy method, which mostly depends on singular value decomposition, can be used in different sciences. I mean, it is not only that in community ecology or in biological data we use principal component analysis. It can be used in any data for reduction of dimensionality. So PCA, there is a unique solution to the eigen analysis. The axes in PCA are called principal components. They are orthogonal to each other. So orthogonal is a mathematical term. So I'll not go into the detail, but you should know that it's orthogonal, and each PC is associated with an eigenvalue. The sum of the eigenvalues will equal to the sum of the variance of all the variables in the data set. So when you have PCA and when you have all your PCs, the first principal component will have maximum explained variance, and then subsequently the second one is bit lower to the first one, the third one is bit lower to the second one, and you will go on. And simultaneously, if you have an N cross P matrix, you have a P cross P number of PCs, and the sum of all these PCs will be equal to 100%, and they will explain all the variances. But in most cases, what we do, we plot only PC1 and PC2 with each other. Axes are ranked by the eigenvalues that I have told you. So they have a direction, they have a value. The first value is the first eigenvalue corresponds to the first PC, and the second eigenvalue corresponds to the second PC. There are potentially large number of axes that I have told you. It depends on the number of, it depends on the dimension of your matrix. Species and samples are coordinated simultaneously. In most applications of PCA, variables are often measured in different units. So that is very important. Like when you do PCA, you have to do a kind of scaling of your data set. Unless you do the scaling of your data set, your PCA can also give some biased output. For example, let's say you have plant distribution data where you have counts. So you say that species A is 1, species 2 is 0, species 3 is 5, species 10 is species 10 is, let's say, 9. So here all of your values are in 1, 2, 0, 5, 10. in that scale. But if you link that, if you link that with elevation, and the unit of your elevation is in let's say meters, it can be 4,000, 5,000, 8,000, 9,000. So there is a mismatch between the scale of your data. So when there is a mismatch between the scales of your data, what you need to do, you need to do a normalization of your data set. That is a very important part of PCA analysis. Other than that, if you do PCA, your results could be a bit kind of, I mean, it could be in a way, I mean, it would be hard for you to analyze. When we do PCA, there are many methods. I mean, there are many tools for doing PCA. Earlier, when I was first, I mean, I was an MSc student, we used to do PCA using some Fortran tools. Because these days, you can use MATLAB, you can use R, you can use Fortran. But in R, community data analysis are being done using a package called Regen, where there are many functions like RDA, PRComp, PCA, and R. So these are the different functions that can be used for principal component analysis. And just briefly about the method, the mathematics of this part, I'll just briefly tell that if you assume that this is your community data and this 2, 3, 5, 7, 9, it shows the counts at one location, and 14062 is the count of the same species for another location. What you do first, you calculate mean, so you minus the mean value, and then you calculate the covariance matrix. Once you calculate the covariance matrix, you come up with the your characteristics equation where eigenvalues are calculated, and then based on that, you do a single level equations, you solve this equation, you come up with eigenvalues, and the matrix eigenvalues are with you, and then you come up with a matrix eigenvector. So once you have these eigenvalues and eigenvectors, you have multiple center data by the eigenvector matrix U, and to obtain the position of the objects in the PCA, so you ultimately this state is formed. So once you have this following step, here you have this U. So matrix U contains direction cosines, which are cosines are the angles between the variables and the PCA axis, and finally you come up with the rotated matrix, where these are the principal components, and then you plot them in a 4x4 axis, and then link with variable 1 and variable 2. So this is how you carry out your principal component analysis, and you come up with a plot which is comparatively easier to explain. So what are the steps when you do PCA in R? So you simply use this RDA function, and this code is basically using the Wagon package, where the var space is the data where species distribution data is available. Once you have this PCA, your scaling may be true in case of unmatched scale data. What I told you before, you need to do this scaling for unmatched scale, and then once you do the PCA, you do the bar plot. This bar plot gives you an idea the relative weightages of eigenvalues, and then you calculate sums, calculate the present percent of variance explained by the first two axes, and then finally you plot PCA for site, and you plot PCA for your species. So this is how your data actually looks like. For each site, these are the relative abundances, and these names are actually showing the number of species that is available. So once this calculation is done, you come up with your final species, your final plot, where species versus site plots are available. This is how you do PCA in R. The next part is non-metric multidimensional scaling, and NMDS, when you do non-metric multidimensional scalings, NMDS attempts to represent the pairwise dissimilarity between objects in a low-dimensional space. Again, I told you before, the entire objective of ordination analysis is to bring the dimensionality to a lower dimension. So in NMDS, the non-metric multidimensional scaling is again an ordination method where you bring the dimensionality of your data and you try to visualize the low dimension data as easily as possible, and it makes your data interpretation easy. NMDS is an iterative algorithm. Any dissimilarity coefficient or distance matrix may be used to build the distance matrix used in the input. So basically, NMDS depends on estimation of distance. So what is distance? Distance is basically Euclidean. In most cases, it is Euclidean distance. That means distance from one point to another point. But In NMDS, it is, I mean, once you calculate distance, after that you calculate, you follow the rank approach. So there is no unique ordination results. It is not that, it is not similar to PCA. Like when you do PCA, you your first principal component will have higher explained variance. Then your principal component 2 will have the second highest explained variance. But for NMDs, it is not like that. It is more of user defined. So you have to define from the beginning how many dimensions you want and how you want to reduce them, and then you plot them all together. The number of dimensions of the low dimensional space must be specified before running the analysis. Now, just briefly, again, I tried to not put much mathematics in it, but MDS, multidimensional scaling, is a family of different algorithms, each designed to arrive at optimal low dimensional configuration. So when your dimension is 2 or 3, we call it a low dimension. But if it is more than 3, it is very hard to visualize. I mean, you cannot simply see the variation of the data within 4-dimensional space. It's easier to visualize the data on a 2 or a 3-dimensional space. Typically, MDS can be of 2 types. One is classical MDS and the other is metric or non-metric MDS. In a classical MDS, similarity or similarity in terms of proximity. Proximity is calculated in terms of distance, are defined any pair of objects in space. In mathematics, a distance function that gives a distance between 2 objects is called metric. So it has to follow some criteria, and based on this criteria, you calculate, you say whether you want to use metric classical MDS or non-metric MDS. Given a set of dissimilarity, one can ask whether these values are distance, or moreover, whether they can be interpreted as Euclidean distances. I'll not go into the details. So this is the basic fundamentals of non-metric multidimensional scaling. And when you use classical MDS, what classical MDS does, it seeks to find an optimal configuration xi that gives you a distance like this as close as possible. For non-metric MDS, given a low dimensional P is nothing but a matrix, and non-metric MDS seeks to find an optimal configuration where x belongs to a rational number that gives you a distance which is again as close as possible. The difference between a metric and a non-metric multidimensional scaling is the classical multidimensional scaling tries to reproduce the original metrics or distances, whereas on the non-metric multidimensional scalings, it assumes that only ranks of the distances are known. The distance themselves are not reproduced. So when we do in MDS, we come up with ranks. It is more user-friendly when you have different parameters from different sources. So non-metric MDS, because it uses rank rather than using the basic distance, because it uses rank, it is easier when you have parameters from multiple different sources. Now, how do you use an MDS in R? Again, I mean, you use this Wagon package in R. First of all, you calculate distance. So these distances, in most cases, we calculate break artist distance. There are many methods of calculating distances between points. So in first step, you calculate distance, and then you use a customized function to plot stress versus dimensionality. And when you plot stress versus dimensionality, it gives you a plot like this. So the ultimate aim is to reach to a dimension where your stress is minimum, but you have to pick up the first hinge point. So in this diagram, you can see that your first hinge point is at this dimension number 2. So your preferred dimension for NMDS would be 2. In case for your data it comes to 3, you can use your preferred dimension for NMDS would be 3. But here for this, in this diagram, what the space and number dimension, you can see from this plot your preferred dimension is 2, where you get the first hinge. And after that, once you have identified your dimension, you use a meta MDS program within R, and using this meta MDS, you calculate NMDS. So meta MDS is a function where you find it in R Wagon package, which can be applied, which applies the square root transformations on the community matrix. Meta NMDS doesn't do that, so there is no transformation. So we use meta NMDS. This method would not give you species score, hence used in next function. So after you have this NMDS data with you, you plot sites and species. So first you plot the similarity of sites, and then you plot similarity of species. So when you plot sites and species, you get this kind of diagram. So these names in reds, they are showing names of species. So basically, with this diagram, what you explain that how similar the species are. So those species which are very close to each other, they have very similarity, and those species which are completely far away from each other, they have highest dissimilarity. For example, this species, it has highest dissimilarity with species number this. Now, when you overlay the site over it, these 1, 2, 3, 4 numbers, 27, 28, these numbers are actually showing the sites. So when you overlay your site, these sites, number 21 sites is actually showing affiliation to species this, species Dichropoly, species this. So when you overlay your site with your species, you come up with similarity assessment. We have now a nice ordination plot, and we know which plots have a similarity species composition. We also know that the first ordination axis corresponds to the largest gradient in our dataset. The second axis, the second biggest. Now, when you have the site and species similarity plot with you, on a third diagram, you can also plot environmental parameters. Now, to do environmental parameters, you can read your environmental data set. Your environmental data set should also look like this. You put all the environmental parameters as columns and your sites as rows. And once you have all these environmental parameters with you, you use the NVFig functions in R. Using this NVFig function, you can calculate NDS, and then finally you can plot the environmental parameters. And once you plot the environmental parameters, you can again come up with a similarity assessment, that what site is having similarity with what environmental parameters. For here, for this site, let's say site number 3 is having affiliation to aluminium and iron. For example, site number 27 having affiliation to calcium and magnesium. So this is how you use non-metric multidimensional scaling for coordination, for ordination methods and for community ecological data assessment. But again, as I told you before, these methods are equally applicable to hydrological as well as meteorological data analysis. I'm using non-metric multidimensional scaling for some of the water chemistry data with us, where we have water samples collected from different springs and for which chemistry data is available with us, and we are using multidimensional scaling to find out similarities. What type of spring is having similarities in terms of chemistry, in terms of discharge, and in terms of geological parameters. I hope I have completed by 4:30. So thank you again. Now, if any of you is having any questions, I'll be happy to answer. Thank you. हेलो
हेलो सर
हाँ थैंक यू अगर किसी को कोई क्वेश्चन है तो आई बी हैप्पी टू आंसर
thank you sir for your this nice talk on this topic climatology and flora and fauna. So I just open this session for the participants to ask for any doubts. So as I am being telling before, you can unmute yourself and ask so that others also can listen. So there were specific, actually, it was really cooling my eyes. Something sir talked about mathematics, and whenever he told that he has cut short the mathematics, I was a bit disappointed. I was really happy. So principal component analysis, all this I have been studying. So especially these are parts of machine learning. So now people can connect. So the machine learning, deep learning, the peoples are fancied by these words. Actually, these are parts of machine learning.
yes.
Yeah,
Yes.
all the things which we have been going through actually connects with these topics, this machine learning and deep learning. So you may ask questions to sir. Yeah, please, participants, please. in the chat window. Yeah, there's a question, sir. What could be the?
So I just wanted to know.
Yeah, yeah,
Sorry.
yeah. Please go ahead, sir. Go ahead.
What could be the applications of PCA in medicine?
Yeah.
See, I mean, PCA is mostly used for trend for basically spatial trend assessment. So what we do for our analysis, we use PCA for for trend analysis to see how the dimensionality can be reduced, and at what component you are having the maximum explained variance. So for example, if you have uh, let's say uh, if you have medicines tested with multiple uh, you have, let's say you have different doses, and the outcome of those particular doses for your patients. You have, let's say, 50 number of different 50 number of different patients, and they have different conditions, and with those patients you are having 100 ml of certain medicine, then 200 ml of certain medicine, or let's say 50 ml of certain medicine. So with your experiments you come up with a database where your rows are actually showing patients having different conditions, and where your columns are actually showing loadings. Loadings means what are the medicines that you are, what are the concentration of medicines that you are giving to your patients. Now, when you do PCA upon such data set, you come up with an optimum. The PCA1 will show you the optimum direction. So, and that optimum directions can be used to find out what dose is suitable for what kind of patients. So I think that can be one type of applications of PCA in medicine. And then again, the best thing is to go and search in Google Scholars for application of PCA in medicinal science. You probably find a lot of manuscripts where different experimental designs are elaborated, and you can follow some of the experimental designs in your work. I think, I think, uh, that
Yeah, yeah,
that's all
yeah.
from me.
Yeah, any other participant? Yeah, Jitendra Rajput, sir. Yeah, please.
Yes, sir. So I just wanted to know as to how we can use. I'm currently working as a assistant professor. I'm teaching bioinformatics, and in bioinformatics, we have to predict a lot of times the position of a gene. So how can we use? And I'm learning Python also for the same reason. We want to. I want to develop some kind of a system by which we can uh, predict the position of the gene. So how can we use machine learning or maybe the regression models, etc.? So I'm just a beginner in that field. That's why I just wanted to know as to how we can go ahead with that.
Yeah,
All
I
the
think.
models, because we have to. Yeah.
Yes, I think prediction is a bit different. I think in that case, probably you need to. Uh, probably I don't know about the schedule of the course. Most probably, some experts will present on predictions. So there are many methods for predictions. Like regression is the simplest method of prediction, and there are many variations within regression itself. So you can have linear regressions, non-linear regressions, multiple non-linear regressions, and then there are many methods for predictions. So even artificial neural network is a machine learning tool which can be used for
Yes.
prediction.
Yes.
Uh,
Yes.
uh, so uh, it's more of uh, uh, I think it will be suitable for someone to reply. I think
Okay.
Professor Dr. Kumar can answer you in detail probably on a session
Okay.
where prediction is discussed. Right now, I think we
Okay,
haven't
sir.
discussed on the prediction
Okay.
thing.
Okay, sir.
Yeah.
Yeah. Thank you.
No, prediction means in the sensor. What do you want to predict actually? No,
Actually, I just want to feed data about various different types of genes to my program, or maybe some sequences, and using those sequences, some consistent sequences and express sequence tag. I just want to feed it to the program so that it identifies if there is, let's say, a specific express sequence tag, then there's a high probability that it's a gene. So I'm thinking on that. aspect?
Sir, actually, we are not experts, but you can pose this question to our experts.
Sure,
At the
sure.
same time, I can refer you some books like you can just type in Google. You can get a lot of books. Maybe you may be aware of these books also, but still I am
Okay, sure,
suggesting
sure.
by my maybe publisher is O'Reilly,
Okay.
Bioinformatics using Python. Or you just
Yes.
type bioinformatics using R.
Yeah.
You are having.
I have that
Yeah.
bioinformatics you are using Python, and
Yes,
I'm
sir. Yes, sir.
referring that. So I just
Yeah.
wanted to know if people are working on that and on the same concept. So I just wanted to get an idea. Anyways, thank you so much. I'll
No,
definitely
no.
answer any other
Actually,
questions.
no. Actually, you put up this question tomorrow to the IIT
Sure,
experts there,
sure,
because
sure,
definitely their students will be working and they may be having some
sure,
directions.
sure. Thank you so much.
Mm, yeah, sir. Yeah. Any other participant who would like to?
Yeah, one Debashish Mahapatra, he asks that please suggest some open sources for ecology data sets. See, what I have presented in R, there is an open source data which is in Wagon package. So you go and download the Wagon package, VEGAN. It's a package that is available within R, and that package has open source data. It has open source data on species distribution as well as species composition. So that is available with the vegan package in R.
Sir, one more question. Actually, um, sir, can we do this things, these operations, all these data sets using our home PC, or what is the finance, minimum finance, which we need to?
It is all available. Actually, see, I mean, I'm also doing all my R analysis using my own laptop or even home
Okay.
PC. R is an open source software. You can simply download R from our repository online. It is an open source software. It is a very strong computational tool. And it is purely open access. I mean, completely open access. And then once you have downloaded R, what I would suggest most of you that you should also download our studio. Our studio is also free. It has some paid component, but we don't need that paid component. We can also download our studio, and then you start downloading packages. It's very easy. You have to, the earlier questions where he was asking about bioinformatics tools. There are many packages available within R where bioinformatics analysis can be done. So I would suggest it can. And the other thing is, R can be installed in a Linux system, in a Unix system, in a I operating systems, as well as normal Windows operating system. So it is it is completely free of cost, and already built-in verified packages are available. So we can use them, as easy as possible. You don't need to worry about the authenticity of packages.
Okay, sir. Okay. So, sir, yeah. Sir, any particular book, say for example, the basic book. You actually suggested two methods: principal component method, and then the other one, multivariable method. The two components you discussed today. So like
Yes.
that, there may be a lot of such techniques, sir. So if we want to study those techniques, one book you suggested that was 1982 published
Yeah, that is
book.
the basic book on, whatever I have suggested, it is mostly associated with the community ecology and data interpretation methods. But other
Okay.
than that, if people are interested for inferential statistics, there are many basic statistical books for multivariate analysis, regression analysis, machine learning methods. So for us here, for our group where we do hydrology and meteorology, there is one book that is kind of basic handbook for us. It's called Statistical Analysis for Atmospheric Processes. It is by Wilkes. So
Yes, sir. Just a minute, sir. I will just type in so that the participants can get it. Statistics analysis.
it is Statistical Methods in Atmospheric Sciences.
By, sir? By?
In Atmospheric Sciences. I'm typing it down.
By. Author name, sir?
By D. Wilkes. It's international. So I think, although it is for atmospheric sciences, but anyone can use that book. It is very handy, and it's a complete, complete description of basic statistics that what we can use for all of our data analysis.
Sir, one more question. Sir,
Yes.
normally in data science, what we have is any data science book. I have in my desk itself, I have one book. I'll just read out the topics. So if we study this and understand this, will it be okay for continuing further? Like you have machine learning, descriptive analytics, then they suggest probability distribution and hypothesis, linear regression,
Yes.
then classification problems, advanced machine learning techniques like K clustering,
Yeah.
lasso, elastic methods, clustering techniques, then forecasting. Is this okay to start any research?
Yeah, I mean,
So,
see, personally, for me, it is. I always tell to our students here that before using a statistical methods or a statistical tool, we should come up with a problem. I mean, what problem we want to address, and that is how you come up. Once we have finalized that our problem is this, then we can come up with a statistical method which is suitable for it. So there are many methods available, many different types of methods. I mean, these days people are using SOMs, people are using artificial neural network, fuzzy logics. I have also seen, I have also reviewed one or two manuscripts on swarm intelligence. Swarm intelligence is very much like how the ants move in a group. So people are using swarm intelligence method for atmospheric processes. And then I have recently I have to review a manuscript where two different algorithms people are using. One algorithm is a gray wolf algorithm. It's more of like copying how a pack of gray wolf moves, and they attack a prey and identify a prey and attack a prey. So there are many such algorithms, there are many such methods available these days with us. The basic problem is how and when to use those algorithms. If I want to use machine learning or a method of machine learning, let's say artificial neural network is a method of machine learning. You can use PCA, you can use factor analysis. So these days people even use hot maps. Hot maps are nothing but combinations of simple correlation analysis, which is coupled with factor analysis. So before doing such different types of analysis in our data, I think it is a good practice to frame the objective. So if my objective is to predict some phenomena, then definitely two things comes: whether the prediction has already been done, or whatever I am doing is completely new. You can approach the problem from two point of view. One is you come up with a new algorithm, you combine different algorithms together, and you make a new algorithm for predicting something, or you do the prediction for completely a new set of data which has not yet been addressed by anyone. Let's say wavelet based regressions is also available nowadays. I mean, people have done wavelet to some time series data, and then using some wavelet coefficients, they have come up with a regression model, and based on that regression model, they are predicting. So your manuscript can be from 2-3 different points of view. One, it can be purely on a method manuscript. That means you are focusing the method, and the other thing, you may completely focus on the problem. Let's say nobody has ever predicted how the snowfall in the Himalayas have changed over the time, and whether there is any relationship between snowfall decrement with the temperature of Bay of Bengal. So it may be a new problem which you want to address, and then there may be a new method what you want to use. So it is better to frame the objective first, and then to use your methods, rather than rather than trying with multiple multiple techniques, because a lots of publications are available these days. So when we try to frame an objective, I think we should do a thorough review, and then we should start working on it.
Okay, sir. Okay. Thank you, sir. I think yeah, sir. I think there are all questions have been answered, sir.
Yes.
I thank you for coming, as you mentioned, within a short notice. Again, on behalf of participants, I thank you for this wonderful session. So, yeah, Mohanty, sir.
Yes, sir. Yes, sir.
Yes, sir. If you want to say a few words, you
Okay,
can
sir.
just.
Okay, sir. Thank you, sir, Mukherjee sir, for your wonderful presentation, sir.
Thank you very much.
We look forward to having you again for our next series of our workshop on this computational biology.
Thank you, sir, very much. Thank you very much. It's a pleasure for me, and I thoroughly enjoyed. I hope that next time I'll have some more time and I'll come up with a better presentation.
Also, you are expecting, sir, next time that this pandemic situation will be over, we can meet physically in our institute, sir.
Yeah, definitely, sir. It will be really good for us. We will be really delighted if you can come and visit us.
Okay, sir. Thank you very much, sir.
Thank you very much, sir.
Thank you, sir. Yes, sir. Now you can log off.
Log off. Yeah,
So,
I can log off.
yeah,
Thank you very much. Thank you,
sir. So, participants, thank you for participating actively. Thank you. So we will meet tomorrow right at 9 o'clock for the next new session. So thank you participants. Thank you all. And I also thank our session chair, Mohanty sir. Sir Mohanty sir, thanks for coming and gracing the occasion. So thank you, Mohanty sir.